60 carbon dioxide transport. In the body's tissue cells, oxygen reacts with various foodstuffs to form large quantities of carbon dioxide in the tissues. The CO2 enters the tissue capillaries and then is transported back to the lungs. CO2, like O2, combines with chemical substances in the blood that increase CO2 transport 15 to 20 fold. Coming to transport of CO2 in the blood, it is not nearly as problematic as transport of O2 because even in the most abnormal conditions, CO2 can usually be transported in far greater quantities than O2. However, the amount of CO2 in the blood has a lot to do with the acid-base balance of the body fluids. Under normal resting conditions, an average of 4 ml of CO2 is transported from the tissues to the lungs in each 100 ml of blood. Coming to chemical forms in which carbon dioxide can be transported, to begin the process of CO2 transport, CO2 diffuses out of the tissue cells in the dissolved molecular form. Okay? And then on entering the tissue capillaries, it initiates a host of almost instantaneous physical and chemical reactions. So some of it remains as dissolved CO2, that is 7% of it remains as dissolved CO2. And 23% of CO2, it combines with the hemoglobin to form carbaminohemoglobin. And the rest of it, it combines with water to form H2CO3, which then splits up into H plus and HCO3 minus ions. This H plus, it is buffered by the hemoglobin again, and then HCO3 minus ions, they are exchanged for chloride ions. This is called chloride shift. Okay, so this is in brief a diagram that summarizes all these things. Coming to transport of CO2 in the dissolved state, a small portion of the CO2 is transported in the dissolved state to the lungs. Recall that PCO2 of venous blood is 45 and that of arterial blood is 40 mm of Hg. The amount of CO2 dissolved in the fluid of blood at 45 mm of Hg is about 2.7 milliliters per deciliter, 2.7 volume percent. The amount dissolved at 40 mm Hg is about 2.4 volume percent, and there is a difference of 0.3 ml. Therefore, only about 0.3 ml of CO2 is transported in the dissolved form by each 100 ml of blood flow. This dissolved form is responsible for adhering to Henry's law. This is about 7% of all the CO2 normally transported. Okay. Coming to transport of CO2 in the form of bicarbonate ion, the dissolved CO2 in the blood reacts with water to form carbonic acid or H2CO3. This reaction would occur much too slowly to be of importance were it not for the fact that inside the RBCs is a protein enzyme called carbonic anhydrase, which catalyzes the reaction between CO2 and water and accelerates its reaction rate about 5,000 fold. The reaction occurs so rapidly in the RBCs that it reaches almost complete equilibrium within a small fraction of a second. This phenomenon allows tremendous amounts of CO2 to react with the red blood cell water even before the blood leaves the tissue capillaries. Coming to dissociation of carbonic acid into bicarbonate and hydrogen ions, in another fraction of a second, the carbonic acid formed in the red blood cells, that is H2CO3, dissociates into H plus and HCO3 minus ions. Most of the H plus ions then combine with hemoglobin in the RBCs because the hemoglobin protein is a powerful acid-base buffer. In turn, many of the bicarbonate ions diffuse from the RBCs into the plasma, while chloride ions diffuse from the RBCs into the RBCs to take their place. This diffusion is made possible by the presence of a special bicarbonate chloride carrier protein in the RBC membrane that shuttles these two ions in opposite directions at rapid velocities. Thus, the chloride content of venous blood cells is greater than arterial red cells, a phenomenon called the chloride shift. Okay. Coming to this point, the reversible combination of CO2 with water in the RBCs under the influence of carbonic anhydrase accounts for about 70% of the CO2 transported from the tissues to the lungs. This means of transporting CO2 is by far the most important. Indeed, when a carbonic anhydrase inhibitor such as acetazolamide is administered to an animal to block the action of carbonic anhydrase in the RBCs, CO2 transport from the tissues becomes so poor that the tissue PCO2 may rise to 80 instead of the normal 45 mm of Hg to get the optimal pressure gradient to cause CO2 transport. Coming to transport of carbon dioxide in combination with hemoglobin and plasma proteins or carbaminohemoglobin, in addition to reacting with water, CO2 reacts directly with amine radicals of the hemoglobin molecule to form the compound carbaminohemoglobin. This combination of CO2 and hemoglobin is a reversible reaction that occurs with a loose bond so that the CO2 is easily released into the alveoli, where the PCO2 is lower than in the pulmonary capillaries. A small amount of CO2 also reacts in the same way with the plasma proteins in the tissue capillaries. This reaction is much less significant for the transport of CO2 because the quantity of these proteins in the blood is only 1/4 as great as the quantity of hemoglobin. The quantity of CO2 that can be carried from the peripheral tissues to the lungs by carbaminohemoglobin combination with hemoglobin and plasma proteins is about 30% of the total quantity transported. That is normally about 1.5 ml of CO2 in each 100 ml of blood. Okay, however, because this reaction is much slower than the reaction of CO2 with water inside the RBCs, it is doubtful that under normal conditions this carbamino mechanism transports more than 20% of the total CO2. Okay, that is why we say it is approximately 23% that is transported in combination with hemoglobin. Coming to the diffusion of CO2 from the peripheral tissue cells into the capillaries and from the capillaries into the alveoli, when O2 is used by the cells, virtually all of it becomes CO2 and this increases the intracellular PCO2. Because of this high tissue cell PCO2, CO2 diffuses from the cells into the tissue capillaries and then is carried by the blood to the lungs. In the lungs, it diffuses from the pulmonary capillaries into the alveoli and then it is expired out. Thus, at each point in the gas transport chain, CO2 diffuses in the direction that is exactly opposite to the diffusion of O2. Yet there is one major difference between diffusion of O2 and CO2. CO2 can diffuse about 20 times as rapidly as O2. Therefore, the pressure differences required to cause CO2 diffusion are, in each instance, far less than the pressure differences required to cause O2 diffusion. The CO2 pressures are approximately the following. See, the intracellular PCO2 is about 46 mm of Hg and the interstitial PCO2 is 45 mm of Hg. This 1 mm of Hg pressure differential is enough for CO2 to enter the interstitium from the tissues. Okay. Then PCO2 of the arterial blood leaving the tissues is entering the blood tissues is about 40 mm of Hg and PCO2 of venous blood leaving the tissues is 45. Okay, so the CO2 travels through this gradient as well. So the tissue capillary blood, as you can see here, it comes almost exactly to equilibrium with the interstitial PCO2 of 45. PCO2 of the blood entering the pulmonary capillaries at the arterial end. See, if pulmonary veins are like arteries and pulmonary arteries are like veins, right? So at the arterial end, it is about equal to venous blood, that is 45 mm of Hg. PCO2 of the alveolar air is 40 mm of Hg. Thus, only a 5 mm of Hg pressure difference causes all the required CO2 diffusion out of the pulmonary capillaries into the alveoli. The PCO2 of pulmonary capillary blood falls to almost exactly equal to the alveolar PCO2, as you can see here, which is 40. Okay, when before it has passed more than about 1/3 the distance through the capillaries. For oxygen, if you remember, it was like this. For CO2 it is the reverse, like this, but it occurs in almost 1/3 of the distance it courses through the capillaries. Coming to the effect of rate of tissue metabolism and tissue blood flow on interstitial PCO2, a decrease in the blood flow from normal point A here to the point B, a decrease in the blood flow causes the peripheral tissue PCO2 to increase from the normal value of 45 to an elevated level of 60. Conversely, increasing the blood flow to 6 times the normal, that is point C. Okay, increasing the blood flow to 6 times the normal decreases the interstitial PCO2 from the normal value of 45 to 41 mm of Hg down to a level almost equal to the PCO2 in the arterial blood entering the tissue capillaries. We need to remember that as blood flow increases, it washes out the carbon dioxide that is formed in the tissues. So that is why PCO2 decreases if blood flow increases, and the converse is also applicable. You also need to note that a 10-fold increase in tissue metabolic rate greatly elevates the interstitial fluid PCO2 at all rates of blood flow, whereas decreasing the metabolism to 1/4 of the normal, it causes the interstitial fluid PCO2 to fall to about 41 mm of Hg, okay, and closely approaching that of the arterial blood, that is 40 mm of Hg. The curve shown in this figure, okay, is called the carbon dioxide dissociation curve, and it depicts the dependence of total blood CO2 on all its forms of CO2. Okay, all its forms on PCO2. We need to note that the normal blood PCO2 ranges between a normal level of 40 to 45 mm of Hg in arterial blood and it ranges between 40 mm of Hg in arterial blood to 45 in venous blood. We need to note that the normal concentration of CO2 in the blood in all its different forms is about 50 volume %. Okay, and which corresponds to the normal range here, 50 volume percent. But only 4 volume percent of this is exchanged during normal transport of CO2 from tissues to the lungs. That is, the concentration rises to about 52 volume percent as the blood passes through the tissues and falls to about 48 volume percent as it passes through the lungs. When oxygen binds with hemoglobin, carbon dioxide is released. This is called the Haldane effect to increase CO2 transport. See, earlier in this chapter, we pointed out that an increase in CO2 in the blood causes O2 to be displaced from the hemoglobin, and this is called the Bohr effect. Okay, the reverse is also true. Binding of oxygen with hemoglobin tends to displace CO2 from the blood. This is called the Haldane effect, and it operates at the alveolar level. The Bohr effect operates at the tissue level. The Haldane effect is quantitatively more important in promoting CO2 transport than is the Bohr effect in promoting O2 transport. Okay, so when coming to the Haldane effect, it results from the simple fact that combination of oxygen with hemoglobin in the lungs causes the hemoglobin to become a stronger acid. This displaces carbon dioxide from the blood into the alveoli in two ways. The more highly acidic hemoglobin has less tendency to combine with CO2 to form carbaminohemoglobin, thus displacing much of the CO2 that is present in the carbamino form from the blood. The increased acidity of the hemoglobin also causes it to release an excess of hydrogen ions, and these bind with bicarbonate ions to form carbonic acid. This then dissociates into water and carbon dioxide, and the carbon dioxide is released from the blood into the alveoli and finally expired out into the air. Okay, and see, this figure, it demonstrates quantitatively the significance of the Haldane effect on the transport of CO2 from the tissues to the lungs. It shows small portions of two CO2 dissociation curves when the PO2 is 100 mm of Hg, which is the case in the blood capillaries of the lungs, and when the PO2 is 40 mm of Hg, which is the case in the tissue capillaries. Okay. Point A here, it shows that the normal PCO2 of 45 mm of Hg in the tissues causes 52 volume percent of CO2 to combine with the blood. Okay, on entering the lungs, the PCO2 falls to 40 mm of Hg and the PO2 rises to 100 mm of Hg. If the CO2 dissociation curve did not shift because of the Haldane effect, the CO2 content of the blood would fall only to 50%, which would be a loss of only 2 volume percent of CO2. However, the increase in PO2 of the lungs lowers the CO2 dissociation curve from the top curve to the lower curve of the figure, so the CO2 content falls to 48% volume percent. This represents an additional 2% volume loss of CO2. Thus, the Haldane effect approximately doubles the amount of CO2 released from the blood in the lungs and approximately doubles the pickup of CO2 in the tissues. Coming to change in blood acidity during CO2 transport, the carbonic acid formed when CO2 enters the blood in the peripheral tissues decreases the blood pH. However, reaction of this acid with the acid-base buffers of the blood prevents the H+ concentration from rising greatly. Ordinarily, arterial blood has a pH of about 7.41, and as the blood acquires CO2 in the tissue capillaries, the pH falls to a venous value of about 7.37. In other words, a pH change of 0.04 units takes place. The reverse occurs when CO2 is released from the blood in the lungs, with the pH rising to the arterial value of 7.41 once again. In heavy exercise or other conditions of high metabolic activity, or when blood flow through the tissues is sluggish, the decrease in pH in the tissue blood can be as much as 0.5, about 12 times the normal, thus causing significant tissue acidosis. Coming to changes in blood gases during hypo and hyperventilation. Blood gas measurements can be monitored either using non-invasive methods or using invasive methods. Invasive methods include the measurement of blood PO2, CO2 and pH. The arterial blood gas ABG measurement is one of the most commonly used techniques for measuring PO2, CO2 and pH and bicarbonate concentration. This measurement technique requires an arterial sample, blood sample of a few ml, and is highly reliable and reproducible. Coming to hypoventilation, it may occur if the respiratory centers are affected by injury or are depressed due to various other reasons. Some of the causes include damage to the nerve supply of the respiratory muscles, spinal cord injuries, conditions such as myasthenia gravis that affect the neuromuscular junction, and dysfunction of the lung mechanics such as chest wall injuries, kyphoscoliosis, and airway obstruction. In all those individuals who ascend to high altitudes, alveolar hypoxia or hypoxic hypoxia may occur due to decreased barometric pressure at higher altitudes. Both hypoventilation and ascent to high altitudes cause alveolar hypoxia, decreased arterial PO2, and increased CO2 levels in the blood, which is also called hypercapnia. Ultimately, hypoventilation will lead to respiratory acidosis, and this is compensated by renal HCO3- retention and excretion of H+ ions by the kidneys. Acid-based nomograms allow simultaneous plotting of pH, CO2 and HCO3 minus concentrations and help in understanding and interpreting mixed respiratory and metabolic acid-based disturbances. Coming to hyperventilation. Hyperventilation is a condition where the rate of alveolar ventilation is increased more than the rate of CO2 production by the metabolic processes. This leads to a decrease in arterial PCO2 levels well below the normal PCO2 range, which is 37 to 43 mm of Hg. Conditions such as tachypnea, which is an increase in the respiratory rate, and hypernia, which is an increase in minute ventilation, should be differentiated from hyperventilation. Hyperventilation causes a decrease in the arterial PCO2 levels, but tachypnea and hypernia do not cause changes in the arterial PCO2 levels. The causes of alveolar hyperventilation could be due to an excessive ventilatory drive caused by either behavioral or metabolic regulatory systems. Some of the causes for hyperventilation include hypoxemia caused by pulmonary diseases, pneumonia, cardiovascular diseases such as congestive cardiac failure, and metabolic diseases such as acidosis, hepatic failure, neurological causes such as anxiety, in which you hyperventilate, and drugs such as salicylates, beta-adrenergic agonists, and miscellaneous causes such as fever, sepsis, and pregnancy. In conditions where the arterial PCO2 is very low due to hyperventilation, it may temporarily cause apnea because of the reduced ventilatory drive. CO2 that is produced by metabolic processes will then build up and restore the ventilatory drive. Chronic hyperventilation leads to respiratory alkalosis, and this condition is compensated by the renal retention of hydrogen ions and excretion of HCO3 minus by the kidneys. Finally, coming to respiratory exchange ratio, normal transport of O2 from the lungs to the tissues by each 100 ml of blood is about 5 ml, whereas normal transport of CO2 is from tissues to the lungs is about 4 ml. Thus, under normal resting conditions, only 82.5 as much CO2 is expired from the lungs as O2 is taken up by the lungs. The ratio of CO2 output to O2 uptake is called the respiratory exchange ratio, or R. Okay, the value of R changes under different metabolic conditions. When a person is exclusively using carbohydrates for body metabolism, R rises to 1 from 82.5. 0.825. Okay. Conversely, when a person is using exclusively fats for metabolic energy, the R level falls to as low as 0.7 from the normal 0.825. The reason for this difference is that when O2 is metabolized with carbohydrates, one molecule of carbon dioxide is found for each molecule of O2 consumed. And as the numerator increases, the R increases to 1. Whereas when O2 is reacted with fats, when O2 reacts with fats, a large share of the oxygen combines with hydrogen atoms from the fats to form water instead of CO2. So in other words, when fats are metabolized, the respiratory quotient for the chemical reactions in the tissues is about 0.7 instead of 1. For a person on a normal diet consuming average amounts of carbohydrates, fats and proteins, the average value for R is considered to be 0.825.